Hi, everybody. Welcome to the Chats with Chaudhry. I hope you're safe and well. Today, I'm delighted to be chatting with Biotage and Astrea Bioseparations CEO Frederic Vanderhaegen. And we're going to learn more about Astrea Bioseparations. I had the pleasure of visiting their Canton facility, which is near Boston, last week as part of my on-site with Chaudhry series. So we're going to learn more about Astrea Bioseparations, but also how it fits in with Biotage's overall portfolio. So first of all, Frederic, it's lovely to meet you. How are you, first of all?
Thank you, Rizwan. Good morning, good evening, good afternoon, everyone, depending which where you're joining us. So it's my real pleasure to join this chat with you, Rizwan, and to have the opportunity to talk a little bit about Biotage and Astrea as a company. So nice
Brilliant.
to
Well,
meet you.
thank you very much. And we were talking off camera, but you're actually calling in from Switzerland today. Last week we were possibly going to be talking over Zoom while you're in London, so you're traveling the world at the moment.
Yes, indeed. My family is located in Switzerland, so I have the pleasure to be here today. But yes, as you described well, the world of a CEO is to go around. I pledged to be close to where the business does operate. So I was in Cambridge last week and heading for Uppsala next week, and so that's probably the journey of many of us right now.
Yeah, it surely is. All right. Well, look, thank you for taking time out to talk to me today. Now, the first question before we start talking about Biotage Australia and the business is, for viewers who are not familiar with you, actually, can you give us a very quick overview of yourself and your background and your role as the CEO?
Yeah, that's right. We're very happy to do so. So I'm born and raised in Belgium as an engineer. So I became a Swiss over time, and I would say having 2 degrees in engineering, in process development, chemistry, I always pledged to work in that kind of industry, and I was very fortunate to do that. After I completed my engineering degrees, I worked as a researcher, and that's how I was first exposed to recombinant protein. Believe it or not, that goes a while ago. And from that moment, I joined a company in life science that was called Millipore, very active into the filtration, downstream processing. Happened to work in those and covered many of the business of that company, but one of which of interest today, as we speak about Astrea, was when I managed the western part of Europe for the bioprocessing side of the business. Subsequently, and later on, I moved to the US, lived in Boston area, and I covered the US operations there always for Millipore and I evolved across and I think I worked for many of the large conglomerates you can think about, like the
Yeah.
Danaher, the Merck, Millipore, and most recently Aventor. So I covered both distribution side as well as manufacturing side. Through my careers, I made a small stop out of the life science and tool industries to cover and to follow the leaders of Martin Mados, with whom I worked at Millipore, and we carve out a business out of J&J to in the diagnostic field. But that's a little bit about me. I've joined Biotage 5 months ago, as I truly believe it's a fantastic company that has all what it takes to be successful in the space of discovery, applied market, and downstream processing.
Brilliant. Thank you for giving me that overview. Now, let's talk specifically about Astrea, though. So, for
Right.
people who are not familiar with Astrea Bioseparations, can you give us a very quick overview of who Astrea Bioseparations are and what they do?
Yeah, Astrea is actually a proven leader in chromatography solutions and mostly for biopharma purifications industry. So they're basically three main stream of product solutions for plasma purification, solution for chromatography purification, which is the primary method used for large molecule purification, whether you think about recombinant protein or whether you think about antibodies. And last but not least, we have innovative technologies that we call nanofibers that aim to purify cell and gene therapies workflow. So this is about the resin business. Next to that, we also have developed a stream of columns that we're offering from small scale all the way to production scale to our customers. And very recently, as you went to Canton, you had the pleasure to see our column packing facilities
Sure.
that we have done there. So we offer that as a service to our customers, as well as contract manufacturing solutions, combining ligand from our customers to our support materials, so that we can enable new streams of purifications and chromatographies for our customers.
Right, that's really good. Now, Biotage actually acquired Astrea, I think February last year, so it's a relatively new acquisition. So, um, I suppose the question people would wonder first of all is why did Biotage acquire Astrea. Maybe it'll need you to do a step back, say who Biotage are, because I didn't ask you that question. So maybe
Right.
you can give who Biotage and why did they acquire Astrea, and where does it fit in actually with the Biotage portfolio?
Yes, I think in a very short, Biotage is a public Swedish company listed in Uppsala. It's a city in north of Stockholm. It's a company that is a little bit less than $2 billion, 2 billion SEK of revenue, operating in a very profitable environment. And if I would summarize what Biotage used to be very well known for, is for the flash chromatography. So,
Right,
and flash chromatography is a technique being used mostly for small molecule purification. From there, the company expanded and on the workflow from small molecule synthesizers all the way to evaporation of the solvent that is being used for those molecules. And the company expanded its reach to applied market, offering mostly front-end to mass spectroscopy solutions and sample
right.
prep solution. As the company was looking to further grow, they recognized the need to evolve their portfolio and expand the portfolio outside of small molecules. So I think obviously, if you think about the new modalities that are of interest nowadays into the drug discovery and pharma, you would think about peptides. You would be blind nowadays or deaf not realize the benefit of GLP-1 and its attractiveness it does represent in the industry, but it's peptides, it's oligos, and it's large molecules. As the company was operating primarily into discovery and a little bit on scale up, and they were looking for solutions to expand the portfolio in the large molecule and production scale. Astrea came as a natural fit for the company because it operates into productions and it operates into the large molecule. And they had a very strong footprint into production and well recognized as a production partner. So Biotage would offer nowadays solution to move down and to scale down the solution Astrea has. And Biotage had solution in the discovery phase for this large molecule, and now Astrea would offer a scale up of those solutions. So it's seamless opportunities covering from discovery all the way to production for the large molecule. So that's the rationale for that acquisition.
Brilliant. And you were giving me an example actually, which was really interesting off camera, where you mentioned that Biotage were using resins, but they weren't using Astrea's resins. But when you
Yeah,
started using Astrea, maybe you can tell viewers what the difference was.
yeah. So I think two examples here. One is using the resin from a third party, and we really realized that we can actually use the resins also from Astrea and put them in a device that would basically enable early stage discovery and purification of those large molecule with Astrea resin. But more importantly, we were trying to resolve a problem on plasmid prep, which is very important for cell and gene therapies onwards. Is one of the pain point of the customers,
Hmm.
and we struggled for quite a few times to get the right level of yield in terms of purification and get the removal of endotoxin. And through a seamless partnership between the two businesses, because Astra is very active into the field of cell and gene therapies, we overcome the issues we were having at Biotage by just fostering collaboration and knowledge transfer. And other example are endless here. I think we also operate in front end to mass spec for clinical trials, and
Yeah.
Astra is very well present into the plasma business, so they know how to remove the large size molecules that are less of interest for those who are studying a rare type of biomarker in the bloodstream. So all of these are very a few examples of how the collaboration help the business to get stronger.
Yeah, sure. There's some added benefit there from the acquiring Astrea.
Right.
Now, you touched on it a little bit in your answers there, but what makes Astrea Bioseparation unique within the bioseparation space?
Well, I think the company, as I said, has demonstrated high degree of expertise into the field of bioseparation. I used to say that chromatography is a science and an art. So you have fundamental of science that you need to understand, but at the end of the day, it's about understanding how the molecule of interest that you need to extract and purify interact with the ligand that you put onto your beads, and that required a lot of expertise. The arts piece is the packing. So many of our customers are expert into developing molecules, but less so in terms of purifying those. And because we do that day in, day out, and we have a wealth and we have acquired through that journey a wealth of expertise. And I think we are capable now optimize the packing to make it a very fast and turnaround time. So for all our customers, we are capable to pack columns in less than 21 days from initiations all the way to the delivery. So and I would be remiss if I didn't say that one of our key value proposition reside on the monodispersed resin. So it's very well-controlled bead size that allow a high control scale up throughput and performance of the chromatography. So I would summarize that by having expertise in the chromatography fields, optimized column designs, expertise in the packing of the columns, and also ability to combine multiple ligands to the resins to support best the customers. Those are what makes our expertise unique in the field of chromatography. We also have a fantastic development happening into the nanofibers technology that is very suitable for cell and gene therapies or for any sensitive materials, because the porosity is very high, and therefore the shear stress that is very a critical element for when you talk about cell and gene therapies to maintain the yield and the viability of those cells can be preserved to that materials.
Fantastic. And I have to say, viewers, watch out for a video of me filming them packing a column, because I have to agree with Frederic. Having watched and filmed it, it is very much an art as well to see how they actually do that. You do
That's
need a
right.
lot of skill and understanding as you pack that column. Now, looking back at the last 12 months, from an Astrea Bioseparations perspective, what are some of the biggest milestones do you think the company has had, and what sort of made you proud as a CEO for the business?
Well, I think I won't take credit for that because I joined the company only a
Sure,
few months ago.
right.
But I would say the company has made few things that are very attractive. So first, the company is registered in more than 19 FDA approved processes or EMA approved process. The company has continued expanding its innovation roadmap, launching what we call Astrea Adapt, which is very suitable. It's nanofiber technology to use for cell and gene therapies. We do have that availability in small scale and large scales nowadays to support our customers. And we have established many global partnerships with well-known pharma companies and biopharma companies. So I would be remiss also if I didn't say that we have continued expanding to support our growth by building new facilities, one of which you had the pleasure to visit in Canton, Boston. So
Sure.
that helped us to best serve our customers and partners out of the North American region.
Right. Okay. Now let's look a bit more about the actual bioseparation chromatography industry itself. So,
Yeah.
as a bioseparation company, what do you see as some of the trends, exciting trends, innovations that are happening within the sector today?
Well, I think, as you know, this is a critical step for the purification of large molecule monoclonals and recombinant proteins. And we see that the demand for these kind of techniques continue to expand. So I think customers are looking for productivity gains. And as such, they are looking at partners that help them to gain productivity by outsourcing these activities in like column packing, so they can concentrate on what really matters to them and rely upon our expertise to ensure optimized chromatography. And in that respect, they can get reliable and consistent result from columns to columns. So which is very important. In the biopharma, we talk about QBD, quality by design, and we also say that the process defines the product, right? So if you have a consistent process, highly reproducible and reliable solutions that backed up by good documentation, high standard of quality, you ensure that you can deliver day in, day out a very high level molecules to the patient.
Oh, can I ask you a question about that? Because when I was
Yeah.
watching the column packing done, and as I said, it's very much is an art in terms of how they do it. Where do you see automation and AI maybe coming into not necessarily that process, but in terms of the sector in general for the
I,
future?
I I would just say probably less so in the column packing, but more into the control parameters of how you control your process. So more generally speaking, you talk about PAT or CGT, which are two acronym to demonstrate to explain what the people are trying to
Sure.
connect to control the process
Yeah.
during the downstream processing. And I think gathering those data, and AI will certainly be an enabler for that, making insights from the data you can collect to make your process better controlled and enable a smooth technology transfer. This is where I see the benefit of AI. It's well known that some of the customers will develop a molecule in one part of the world, manufacture it in another part of the world. So ensuring a smooth process transfer
Yeah.
through AI and through a more informing the customers earlier about any deviation through the process will probably be some benefit from AI in process control that I could envision.
Right. Okay. And in terms of Astrea, how do you see the company helping shaping the future of that particular field?
Well, I think we continue developing and expanding our capabilities, as I said early on. So by enabling the customers to outsource the critical element to someone who has the high degree of expertise, and enabling that in a very agile way and a faster turnaround time will enable the customers to develop faster and quicker and cheaper the productions of those molecules. So that it's all about productivity gain into the pharma space. That's how we see ourselves. Also combining the ligand from the customers that they have of interest to our monodispersed beads will enable have to get a better performance. I would be also remiss if I didn't say that we are offering resins that are more environmentally suitable than many others, since they are agarose-based resins. So they are very compatible with the pledge of environmental and sustainability that we do here in the industry nowadays.
Brilliant. Now let's talk more about actually the column packing capabilities. As I said, I was lucky enough to view the column packing suite and see you actually packing a column last week in Boston. But what sets that facility apart from others in the industry?
Well, it's our up in North America that we recognize that there's a lot of demand from that part of the world. A lot of innovation is happening on that part of the world, and we would need to be present there. So it's speeding up our process. It makes us able to pack the color in a very short turnaround time. As I said, I think we are aiming for less than 3 weeks. We can do that in multiple types of columns format, from very small scale with a snap column all the way up to the production scales, and quality is a must. So our site is in compliance with ISO 7 clean rooms facilities, ISO 9001, and it's a state-of-the-art facility that we recently opened. So with very expert subject matter person operating into that facility, and a very strong regulatory and control environment. So I think these are the right to play, and the right to win is about our agility, our ability to pack resin from multiple vendors. We are totally agnostic when we do that. And I think what is important is, again, the proximity and the understanding of our customers as it comes to do that. For doing that, we rely upon the knowledge of our field application engineers that are really truly scientists and people who have done process development. Those are actually same persona and same profile as those to whom we talk to as our customers. So
Yeah,
we have actually internal customers that
sure.
that we can rely upon.
Yeah, well, it's funny, having obviously met them, I realize they are very, very knowledgeable and know what they're talking about. And so you've answered this question, I think, already, but how do you ensure the quality and consistency of the columns produced at the Boston facility?
Well, I think one of the key parameters is the resins we are using, right? I think because we have a very good control on the particle size, we can pack the column in a very systematic way and expect the same results. I think without getting too much into the science, right? I think if you want to have a good chromatogram, you want to have a very nice peak and shape of the peak, and I think you basically need to ensure you don't have channels into the columns, because obviously the molecule will go preferably through the channel, and you want to make sure that all the beads that you have is the same shape, same porosity, same particles, so that when you combine your ligand, you have the same profile of elutions. You
Sure.
don't have big and small beads that would bind more or less, and the molecule has to go through those. So the more narrow your dispersion of particle is, the better the chromatogram will be, which ensure a higher linear velocity through your chromatogram.
Fantastic. Right. Well, thank you for letting me know that. Now let's look ahead though, in terms of the future. So if you're looking over the next 5 years, I mean, what can we expect to see, as much as you can reveal? So whenever I ask this question to anybody, it's always a loaded question. Some people are more willing to share than others, and I can understand why as well. But what can we expect to see from Astrea, particularly in the realm of chromatography and bioseparation?
Well, I think you expect to Astrea as a leading provider in chromatography and separation techniques to continue supporting our customers and adjusting to the market needs. So I think we do see a higher growth in for viral vector purification solutions. So we continue expanding there and innovating into that field. We believe that the demand for custom resins when customers are developing new molecules will be driving, and I think we want to be partners into that. So basically, offering custom resin solutions to our customers and custom resin packing to our customers. Packing solutions that we believe that people will more and more concentrate on their core capabilities and externalizing those. Those are the key drivers. As we think about new solutions, we need to continue innovating in ligand ourselves, but we also innovate with others to add their ligand onto our beads so that we can offer a compelling solution to the customers. And we are at the early days of the nanofibers adoptions in the market. So we believe this is a breakthrough technologies that would help the customers to unlock one of the key issues in cell and gene therapies, which is the yield and subsequently the cost of those therapies. So I think it's a very great inspiration for me as a CEO to enable pharma to purify cell and genes and bring new solution to the world.
Brilliant. And on a much wider scale, so the final question of that is that how do you see the company industry actually evolving over the next 5 to 10 years? But there's been a lot of change, obviously, in the industry over the last 5, 10 years anyway, and this has been quite a traumatic year anyway already for many reasons. How do you see the things changing and evolving in the next 5 to 10 years?
I think it's the fundamentals of that industry remain solid, right? I think people have become elder. People want to still live longer. So all these type of chronic diseases that will be addressed by large molecules are as I need, and we have seen recently with the GLP-1 in the peptide that we are making headways and good progress into that field. So I think broadly speaking, I'll see the industry remaining solid and a promising industry to be in. Yes, there are some ups and downs into the industry, like in many others. I think we have seen a shrinkage into the investments, which mean customers had to be a little bit more focused on which molecules they want to bring through clinical trial. But I see that as a short-term issues. I think the artificial intelligence will also enable to develop new molecules more safe and with a high efficacy. So I think and we've seen that monoclonal antibodies remain the largest therapeutic area, the largest type of molecules for oncology, but many other modalities are coming into play, which as they proceed through the R&D and through the development cycle, I think that will be very promising for the industry overall. So I think and frankly speaking, all these techniques do require gentle separation techniques that are chromatography based or nanofiber based.
Brilliant, Frederic. Well, thank you very much. I will ask you one question because I don't to get the opportunity to sit down with CEO. There's always one question I always like to ask: is that from your perspective as CEO, what's your vision for the company going forward?
I think it's a very good question. We are just in the midst with the team to reframe our visions. I think we are developing and articulating that vision about supporting our customers, obviously, but around our expertise or agility. And it's basically enable the customers to purify their molecules with a high degree of safety. So I think I would be tempted to say let's wait a little bit as we will reveal that vision and that new mission soon to the broader audience. We are still in the midst of refining internally, so I would reserve that to our first employees before we go outside.
Well, I understand that completely. Well, look, thank you for talking to me and taking time out in your busy schedule to give us an overview of Astrea, but also a little bit about Biotage as well. Thank you very much. So viewers, if you'd like to learn about Astrea Bioseparations and Biotage, then click on the links above this video. But also make sure you follow me and check out the videos I've done already about my trip to Boston and to the facility in Canton, where I had a fantastic tour of the facility. But also next week I will be sharing a video of them actually packing a column, which was really interesting for me as a non-scientist to see what they're actually doing. So check out that. And obviously, if you've got any comments based on what Frederic said today, then please feel free to put them in the comments below, because I'm sure Frederic would love to see any feedback you have to what he said today. So Frederic, thank you very much for your time. Enjoy your travels. And viewers, thank you for watching. Until next time, as always, stay well and stay safe. Bye bye.
Thank you.